Welcome students, once again in the human histology class videos. I myself, Dr. Aksh Dubey, professor in the Department of Anatomy, is going to give you an idea of the various histological structures of human anatomy. Well, we have studied a lot of sections in earlier videos: connective tissue, epithelium, muscles. Now we are coming to one of the most important topics of the histology, and that is the gastrointestinal tract histology. It's a very long topic of the gastrointestinal tract, and almost 5 to 10 videos are required itself just to complete the gastrointestinal tract histology. It's very lengthy. So I'm not going to introduce so much details of the gastrointestinal tract. Rather, I will give you the view and ideation and correlating with the function how the cells and other structures are changed at various places. Let's see the gastrointestinal tract, basic idea of how histology of gastrointestinal tract look like. Gastrointestinal tract have a fixed pattern. For example, there is a stratified squamous epithelium. Here we see the muscular mucosa is there, then submucosa is there, and then their muscles layer is present, inner circular and outer longitudinal, and then outside layer adventitia or serosa. So here if you connect all this, then we have mucosa, submucosa, muscularis externa and adventitia / serosa. Now come to the mucosa. In the mucosa, there is an epithelium is present, then there is a connective tissue below that, and then there is a structure which is differentiated into the submucosa with the help of smooth muscle structure. Why we have these structures here? To support the epithelium. Usually the GI tracts have very large structure in the form of villi, in the form of folds, that gives you very sort of absorption and secretion functioning. So from that point of view, we have to understand it that the epithelium has a very changes in thickness. But the common feature is if you accept the esophagus, usually the epithelium is simple columnar epithelium, right? The simple columnar epithelium is tagged as a gastrointestinal epithelium lining the whole of the gastrointestinal tract from inside. But in the esophagus, we also see in upper portion that a stratified squamous non-keratinized epithelium is present. Why it is so? Because in esophagus, whatever we eat, if we first come here, imagine a columnar cells are present in the esophagus. What may happen? These cells have very high chances of getting damaged by our tea, coffee or any edibles which can damage them, and later may convert into the various sort of diseases, maybe a cancer as well. So to prevent that, nature knows this, and what it's doing, it's converting into the stratified squamous non-keratinized variety, which give you strength in this region, as well as it protects you from the any edibles. So into the upper part of the gastrointestinal tract, we see this. Also into the exit near the anal canal, we also see stratified squamous there, and in between hole we have this columnar cells. Columnar cells, if you have seen the video of the epithelium, please go there, and you must be knowing that columnar cells are very good in secretion as well as absorption, both a functioning. So columnar cells are present over there. They are the simple columnar cells are present. That is the first thing. Second thing, in the GIT, food is running, right? And food, it has many structures, components present there. For that, it is smooth flowing. We need intermittently present of the mucus cells. So columnar cells, then there are the many number of mucus cells. That is the pattern we see into the epithelium. This mucus cell number is very important as we go from up to down or toward esophagus, stomach, duodenum, jejunum, ileum, cecum, appendix and large intestine. Their number continuously increases, right? So the ratio in the simple epithelium, columnar epithelium and the mucosa epithelium started decreasing as we go up to down because of the high number of the mucus cells in the lower gastrointestinal tract. These cells in histology looks like a very peculiar inverted cup shaped structures, and goblet cells they are named as, and that you can clearly see is a mucous cell which may present. Now again, GIT is not so simple. If epithelium here is not in a very plain fashion as you see in other part, this epithelium makes a fold, various sort of folds. At certain places it is named as villi, at certain places it is made folds depending on whether the submucosa have entered in it or not. Entered there, if there is, then it is a fold, and if it is not, the submucosa is coming there, then it is a villi. Then this villi might be of various shapes, depending on, for example, into the villi of the duodenum, the mucosa look like a leaves pattern. Jejunum, it looks like a tongue pattern, and ileum, it looks like a finger-like pattern. Though it's not fixed, but in the classical histological text, they are mentioning in such ways. You can make your own acronym for them as well, but it is the classical one in the description as well. That, in another word, they have various shapes, and according to their function, it changes, right? In the upper GIT, always remember, it is digestive more, absorptive less, and in the lower GIT, it is absorptive more, digestive less. So, in the upper part, there is absorption is present, but water is absorption not present. Into the lower part, into the water absorption is very, very high. So, according to the need, according to the function, it changes. I have told you many times, it is not the histology which defines the function, it is the function which defines the histology. Just check the function of the organ, and the histology adjusts itself according to the requirement. Now come to the below that, it is the submucosa. Submucosa is not a very special structure, it is simple. The connective tissue is present here with a lot of blood vessels, lymphatic vessels are present here, nerve plexus minus or obox plexus are present here, meandric, obox, these all are the plexus which are present into the gastrointestinal tract at various levels. They are very, very high number of neurons that can control actually the function, and in various levels of the histological side you can visualize them. In this whole process of secretion, absorption, body needs a lot of ATPs, and ATP needed, of course, blood oxygen is required for that, and for the blood oxygen we need a very high vascular supply into the GIT. For solving that purpose, submucosa has many blood vessels are present. Now we come to the outer layer. Here we see in this muscularis externa, inner circular and outer longitudinal. Remember, this pattern is present if you take a transverse section of the tube. If you take a longitudinal traction, it changes in visualization, though inner is always remains circular and outer is always longitudinally present. But if you take a section in the longitudinal of the wall, it flips. It appears in a different way because of the sectioning. So, one thing we have to clear-cutly say, then we study the intestine into the transverse section, not into the longitudinal section. And in the longer transverse section, inner is circular smooth muscle layer and outer is the longitudinal muscle layer. So, circular and longitudinal both have different function. Inner circular, when it is when they contract, it squeezes the whole of the intestine, and the food bolus is shifted either the up or down slightly. But into the longitudinal fiber, when the contraction wave comes, it propels the food to the longer side or the peristalsis side. So, these two type of the muscle fibers have slight different functioning there, and that propels the food from one direction to the another direction. That is why we see inner longitudinal, outer circular. At certain places like the stomach, we have an additional layer of oblique fibers is present over there. So, that could be added over here. In the last outside, we have adventitia or serosa. The serosa word is not written here. What does it mean? Well, the answer lies into the gross anatomy of this organ. We know that abdominal organs are either covered with peritoneum or not covered with peritoneum. If they are covered with peritoneum, that means it is a simple squamous layer which is present outside, because peritoneum is a single squamous layered structure, and then if you see this, then it is named as serosa. For example, stomach we see the serosa, small intestine we see serosa, But if this layer is not present, we see the patches of the connective tissue of disorganized shape, and that the structure is named as adventitia. So if you take outside or extraperitoneal part or layer or the side of the organ, then we do not see this squamous layer, and it is named as adventitia. So serosa or adventitia depends on what side of the organ you have taken for the histological analysis. So these are the various layers. I hope you must have understood till now the basic layers of the gastrointestinal tract. It is a very vast topic which may be cover another time of the various structures. So I stop here in this video and see you in another video with some another topic of your interest. Thank you.